So this video looks at Carlsson et al. Study. This is your contemporary study for the clinical approach. It is named by the exam board, so you could get asked anything and everything about this. Now, just as a little bit of an introduction to this, there is an argument for the role of glutamate in the production of schizophrenia because glutamate is present even in healthy controls in high quantities throughout the brain, but in very high quantities it can be really toxic. Now, the majority of glutamate is actually stored inside your brain cells, and it only works as a neurotransmitter when it's outside your brain cells. Now, chemicals called glutamate transporters uptake the loose molecules of glutamate and then bind them safely inside the cells so that they don't do you any harm. Now, levels of glutamate can be a bit like Goldilocks with the three bears, because in order to maintain a healthy brain chemistry, you've got to make sure that the levels of glutamate within your brain are just right. If you have too high levels of glutamate, then you're going to end up with hyperglutamaturgia, or too lower levels of glutamate will give you a hypoglutamaturgia. Now, as a neurotransmitter, glutamate is linked to memory and learning, and it binds to glutamate receptors. So, for example, NMDA is a glutamate receptor. These receptors are found everywhere in the brain, which is why it's so important that the glutamate levels are kept low in order to ensure healthy glutamate uptake. Now, Carlsson study. It's really important that you're clear on this. This isn't an investigation like an experiment, it's a review. So Carlsson has looked at all the research that exists on schizophrenia and has summarised it into a review. Now, the focus or the aim of the research was to find out whether the dopamine hypothesis was still relevant and whether research suggested that it was the main explanation for why schizophrenia occurs. However, it also seems from this that other neurotransmitters are just important as dopamine and play just as much of a role in the development of schizophrenia as dopamine does. Now, remembering that this isn't an experiment, so there is not an IV or a DV or a cause and effect relationship. All that was trying to happen here was they were trying to investigate the relationship between schizophrenia and dopaminergic dysfunction, which means not enough dopamine, so the dopamine hypothesis, but also to explore a rival theory, which is the glutamatergic deficiency, so the idea that too little glutamate can also link to schizophrenia. There isn't really a procedure to this, with it being a review, but they refer to a number of studies that use positron emission tomography, so PET scans, so just make sure that you're revising those as well. PET scans are obviously brain imaging techniques where we're injecting radioactive tracers that dissolve into the bloodstream. The tracers carried to the brain, and it basically allows us to get a picture of which brain structures are particularly active at that time. Now, it detects the radioactivity and converts it into a digital image, and then we'll have active areas of red and yellow and less active levels at purple and black. Carlsson's results are split into 7 sections. I do not think you will need to know all 7. I would choose a few results to remember that you know in depth and that you can really talk about and summarise. So I'm going to go through some that I suggest you remember. So the first result related to dopamine. Now, Carlsson explains evidence that supports the dopamine hypothesis, but he does point out some problems with it. So Carlsson says that schizophrenic patients show more dopamine activity than a healthy control group, particularly linked to the basal ganglia. Lurel found that schizophrenic patients in remission, so not having symptoms, had normal dopamine activity. Now, this suggests that maybe dopamine links to the symptoms of schizophrenia and not actually the origins of schizophrenia itself. Carlsson also points out that when patients were taking antipsychotics, that was when they complained the most about side effects when their symptoms were in remission. Now, this isn't actually surprising. If their dopamine activity is becoming normal and then the drugs are causing them to have too little dopamine, then they're going to have symptoms and negative symptoms at this point that make them feel ill. Carlsson also suggests that glutamate has a role. So drugs like PCP, also known as angel dust, and ketamine produce psychotic symptoms, but instead of activating dopamine, they actually stimulate glutamate receptors called NMDA, which we talked about at the beginning. Lodge et al. Claim that glutamate activity at NMDA receptors produces psychotic reactions in rats and humans. The third result is actually pairing dopamine and glutamate together. Now, Carlsson suggests that glutamate actually seems to regulate the behaviour of dopamine, and he describes it as an accelerator by saying that it increases dopamine activity, or it can choose to break it and decrease the dopamine activity. Miller and Abercrombie show that the release of dopamine is increased if glutamate activity is reduced. So you're blocking NMDA receptors, you're reducing glutamate, which actually therefore increases dopamine. The psychotic side effects of PCP, the angel dust that we talked about before, are reduced by a chemical which increases the activity of glutamate but doesn't actually do anything to dopamine. Low levels of glutamate seem to link with both positive and negative symptoms, remembering that dopamine only actually linked with positive symptoms. Carlsson locates this area of the brain by calling it the stratum, which is at the bottom of the basal ganglia, in the cerebral cortex, which includes the frontal lobe, where conscious behaviour happens. Carlsson suggests that there are probably different groups of schizophrenic patients, and he calls these subpopulations, whose symptoms have different biological explanations, so not always the dopamine hypothesis. Some may have glutamate deficiency, and what Carlsson is saying is you can't just do a one-size-fits-all approach to schizophrenia. You need to look at each individual patient and therefore come up with a treatment that is individual to them. A lack of glutamate may cause these patients to have an exaggerated response to dopamine in the postsynapse. So what I mean by that is, even though only normal levels of dopamine are being produced, the dopamine receptors are having an extreme reaction which is down to the glutamate. Researchers need to start looking at the role of neurotransmitters like GABA and neuropeptides in order to be able to further the research into schizophrenia. You also need to be able to evaluate Carlsson in quite a lot of detail. So Carlsson is one of the world's most famous researchers into dopamine and schizophrenia. The studies he cites in his reviews, there were 33 of them, 14 of which he took part in. It's a really representative sample of what was going on at the time. However, that time was 1999, which is 2 decades away now, so the study might be time-locked, which means it only represents things that were going on in 1999. If research has moved on since then it isn't representative anymore of how schizophrenia is diagnosed and thought of today. The Carlsson studies are all lab experiments, most of them on animals, and some of them use modern PET scans in order to be able to draw their conclusions. We know that PET scans and animal studies are standardised and replicable, which actually makes the research reliable. Carlsson cites one study, which was the Laurel et al., that hadn't actually been published at the time and therefore hadn't been peer-reviewed, so you should approach that with caution. The application of this research is massive, and the biggest one is that the development of new antipsychotic drugs to improve dopaminergic drugs that have fewer side effects based on a better understanding of the dopamine pathways, and a new atypical drugs that affect other neurotransmitters such as serotonin and glutamate. Glutamatergic antipsychotics are still in development. These drugs promise to bring relief to people who don't respond very well to antidepressants and antipsychotics that focus on dopamine and therefore don't have the same side effects as current antipsychotics within the field. Carlsson is questioning the validity of the dopamine hypothesis that he himself came up with in the 1960s. He lists some of the evidence that has called it into question, such as the new atypical antipsychotics like chlorpromazine, which reduce antipsychotic symptoms without influencing dopamine. He considers an alternative in the glutamate hypothesis. Now, Carlsson actually argues that both hypotheses may be true and that research should continue into both. And over the last 20 years, most other research has agreed with this. Ethically, there are no ethical issues directly related to these studies. However, many of the studies that are cited are animal studies, which may involve ethical issues since the mice are being injected with drugs in order to be able to induce a psychotic symptom. Other studies involving humans with or without schizophrenia being given amphetamines or PCP that may increase psychotic symptoms can also be questioned ethically, because the participants are drug naive, meaning they don't know whether they're being given a real drug or a placebo. So there's issues there with risk of participants, but also deception.